5 infusions of this synthetic HDL at 1-week intervals. And then on the 6th week, we did another IVUS. And I told the team in the lab that this was sort of, I was almost embarrassed to do the study because I said, this can't possibly work. It's too fast. And we know this disease has been there for decades. We knew that by then from Dr. Tushu's work. So we did it because in science you have to do the experiment or you don't get the answer. And the answer was remarkable. In the people that got placebo, nothing happened. P equals 0.97. But in the people that got the drug, they had very, very substantial regression of their coronary disease in both the low and the high dose. We were just astonished by the finding. So was the world. People tended to overreact to this finding, as I will show you. This is the article in Time magazine about it. And I'll tell you, I'll never live this one down. No respectable scientist ever wants to have their work referred to as Drano for the coronaries, but there it was. And there's a picture of some of these very nice people from Limon Silgarda with their carriers of the protein. November 17th, 2003. It's about 7 or 8% of the volume of the plaque was erased in 5 weeks.
It's once a week for 5 weeks?
That's right, once a week for 5 weeks. One of the things about ApoA1 Milano is it has a longer residence time than native A1. So you give it once a week, which is one of the reasons why it may be better. But anyway, the bottom line is that this was IV. Now, for a number of reasons I can't go into, we weren't able to move to the next step, but we're now back in business. And the next human trial is in early stage planning right now.
They've been able to make it?
They found a way to make it. And they found a way to make it in large quantities by the vatful. And we're going through the toxicology and some of the other testing because it's a new formulation. But we're going to get in.
Is it still the same company, Pfizer?
No, Pfizer bought them, then they spun it off, and the medicines company bought it. It's a long story. But we're working very closely with them to move this into the next rather big phase 2, late phase 2, early phase 3 studies, so stay tuned. What did we learn from this? We learned that the HDL hypothesis is solid as a rock, that the idea that you could regress plaque by increasing HDL is there if you can find the right way to raise HDL. This was profound. This, is why we were very excited by the findings. Even though we couldn't move forward, it gave us the scientific underpinnings to move on the HDL hypothesis. Now, nothing that happens in this world escapes the attention of the U.S. government. And so I got a call a number of weeks after we published this manuscript from the National Aeronautics and Space Administration, NASA. And they said, could I urgently fly down to Houston to the Johnson Space Center? They really needed to talk to me right away. And I, being a dutiful citizen who always does what the government tells me to do, I hopped on a plane and went down there, and I met with all these NASA people. And they said, we really want some of that ApoA1 Milano stuff because we want to put it up on the space station. So if any of the astronauts develop coronary disease, we can kind of melt away their plaque with your liquid Drano. And we also think it would be really good for the Mars mission. And so I had to explain to them that it wasn't ready for prime time yet, but in the process I had a good time, and this is me in mission control. You can see there the space station's up there, and and I have a better picture of me sitting in the mission control seat with the mission controller's hat on. They even let me fly the space shuttle simulator, which was really cool. I crashed it in about 20 seconds, but nonetheless, that was a very funny experience. Well, we've gone on with IVUS, and we have studied a whole series of therapies. I'm going to kind of speed through a little bit of it because I've taken longer to get here on this slide than I wanted, but we studied blood pressure lowering drugs, and we're able to show, in fact, that we could lower blood pressure even within the normal range in people with coronary disease from about 129 to about 124. And in the process, we could both reduce events and reduce the progression of coronary disease. So we were able to establish the relationship between blood pressure lowering and deceleration of disease. Now, this is music to Dr. Roizen's ears because he is a great believer in the role of hypertension and atherogenesis. And, Mike, our data shows that, just like you can never be too rich or too thin, you can never have too low a blood pressure, we think, probably. And very dramatic data. We have another study coming online in another couple of years that takes and extends these data to a new group of people. Now remember that with reversal, we'd gotten the LDL down to about 79. As more powerful therapies became available, we had the ability to go lower. And we wanted to go where no man nor woman had gone before, into the realm of Star Trek, into the realm of really low LDL levels. And so we wanted to explore this region down here well below 79. And we did that with a trial known as Asteroid. It was about 350 patients treated for 24 months with even a more potent statin, rosuvastatin. And in this time, we got the LDL from 130 all the way down to 60.8. That's a 53% reduction. That's a low LDL. Not the lowest we've seen, but a low LDL for sure, on average. And equally importantly, this particular statin seems to have a differential boost to HDL levels. It raises HDL more than other statins. And so we had a 14.7% increase in HDL. Look what happened to the LDL-HDL ratio, 58% reduction. What happened is the disease regressed. This time, we didn't just get to zero. We got statistically below zero. It's modest. It's not as big an effect as we saw with ApoA1 Milano, but it's several percentage points reduced disease. And the p-value for regression is highly significant.
How many months?
24 months. 24 months at an LDL of 60 with a 15% increase in HDL, and we got regression.
Do you have a percent of, does that reduce plaque rupture risk, the inflammatory component of it as well?
These studies don't answer those questions. However, other studies done in parallel, like IDEAL and TNT, have shown that it does, in fact, reduce that very low LDL levels within the treatment range that we're exploring do, in fact, produce reductions in morbidity and mortality. Our studies are much too small to get a morbidity and mortality endpoint. So they're proof of concept, they're hypothesis generating, and they've proven reliable when looked at differently. The question is, not everybody regresses. And one of the really important remaining questions, of course, is that about a third still progress and about 2/3 regressed in the study. If you look at a histogram with 0 being neither progression nor regression. We've got to work on the third that, despite low LDL levels, don't regress. And we're doing that with a lot of new therapies. If you put Asteroid now on a regression plot, you see something about as remarkable as anything, any relationship I've certainly seen in medicine. These are all the various studies, and I've skipped over a bunch of them in the interest of time, is that there is a very tight relationship between the LDL achieved on treatment and the progression of disease. And you see right down here, somewhere in the range of around 70 or so, that line goes below 0. And so when you start to get to sub-70, sub-75 levels for LDL, you start to go negative on average. Not everybody, but on average, you get to regression. And if you get down here to 60, you're definitely in the territory where the majority of people will regress. Lower is definitely better, at least by this measure of disease. We then, with Steve Nichols, who has taken over the laboratory after I became Chair of Cardiovascular Medicine, who's a fabulous scientist. Steve then took all of our trials and put them together in order to get more statistical power. And he comes up with this extraordinary relationship between on-treatment LDL and change in plaque. And again, you can see that relationship with even tighter confidence intervals, that there appears to be no plateau, that the lower you go, and you know, what's the LDL of a 5-year-old, normal 5-year-old, Mike? 50, 45? Probably that's where our species needs to be. And if you get down there, you know, really good things happen. You don't get plaques in your coronaries, or if you have them there, they kind of go away. The other thing that Steve did, which is also rather remarkable, is that he looked at these patients who had very modest increases in HDL, and the curve is reversed. And the more you pushed up HDL, the less disease progression you had. And so we could, in a series of trials, when combined into a meta-analysis type of an analysis, show you that these measures, these targets that we have meaning, and there appear to be no clear thresholds for benefit.
That's actual HDL content rather than
Functionality.
Exactly. Have you looked at the functionality of that?
No. And that's going to be an interesting question. Let me just show you this sort of summarized, and I'm actually going to probably stop around now, even though I got more slides to show you. I was going to show you all of our failures as well as our successes, but you know, because we've had studies, and I want to say something about that before I do show you this slide. We're every bit as proud of those therapies we've tested that didn't work, because if you can figure out what doesn't work and take it off the table, then industry and other scientists can work on the things that do. So the entire class of ACAT inhibitors was killed after we did a study that showed that it not only did not regress disease, it slightly increased progression of disease. And we did a study with torcetrapib, a CETP inhibitor, that was ill-fated. And so we're fine with that. That's why we do research, and that's why we get upset when pharma companies sometimes don't publish their negative results, because you've got to tell people about the things that don't work so that we can focus our attention as a society on the things that do. And so we're just as likely to publish just as quickly a negative trial as we are a positive trial. Let me just show you Steve's rather extraordinary analysis that if you took all of our trials, and if your LDL was above the median and your HDL was below the median for the group, you progressed like crazy. The people who actually regressed were the people who got better than median effects on LDL and better than median effects on HDL. It seemed that you had to get some benefit on both of those parameters to turn the disease around. And so we think that there will be more focus on HDL in years to come. As you know, the biggest problem we have is we don't have any good ways to raise HDL, or not very good ways. We have some lifestyle ways, but they're modest in what they can do. And what we really need are more powerful drugs. They're coming. Hopefully they will work. I'm going to stop there, and I'd be happy to take questions. Thank you so much for your attention.
I have a couple questions.
Yeah.
As the statins got stronger, did it make a difference to liver toxicity?
Yeah, it's interesting. Each of the statins have their own distinct toxicities. And so